Hello, everyone. Today we're going to be taking a look at some histology, specifically focusing on the respiratory system. So let's get cracking. So the first important concept to understand when it comes to respiratory histology is the two different zones within your respiratory tract. So you have your conducting zone, which is responsible for, as the name suggests, conducting or allowing the flow of air into the functional component, which is the respiratory zone. So the conducting zone is responsible for transporting air into and out of the lungs, whereas the respiratory zone is what's called the functional zone, because that's where you actually have gas exchange occurring. That's where gases, carbon dioxide, and oxygen are being exchanged between the lungs and the blood. Let's have a look at the very top of the airway, which starts off with the trachea. The trachea is a bit of the airway that sits in your neck, sorry, and you can tell what it is histologically, usually from the large C-shaped rings that you can see on histology, which mark the cartilaginous rings you can feel even if you palpate your trachea. You can also view on histology the trachealis muscle on the posterior aspect. And also in terms of the cell types, although it's kind of hard to make out, there's ciliated pseudostratified columnar epithelium, ciliated again because it's going to form part of the mucociliary escalator that's going to sweep mucus up and get rid of it through coughing it up and spewing it out. Next up, you have your bronchi. So your bronchi consist of ciliated pseudostratified columnar epithelium. So notice how the same cell type is here. So that makes it already easier to remember. Again, they're still ciliated because they're still forming part of the mucociliary escalator. This time, however, you can make out some smooth muscle because the smooth muscle is going to be responsible for contracting and relaxing to allow the airway radius to change. And if you had a look at our breathing biomechanics video, you would notice that that was important for controlling how much air was coming into and leaving the lungs. Another thing to keep in mind is that you still have cartilage here. However, when it comes to the primary bronchi, so coming straight off the trachea, they still have their full rings of cartilage. But when you go further along and bifurcate further to the secondary and then tertiary bronchi, they instead have more irregular patchy plates of cartilage, which is what you can sort of see here in this diagram, where they don't really form a clear, distinct ring. They just form more patches of cartilage that show up on histology. Okay, and now we come to the bronchioles, and these are simple cuboidal epithelium. So now notice we've gone from pseudostratified columnar to simple cuboidal. So a thinner cell layer for the epithelium. And over here, we have absolutely no cartilage. So you shouldn't see any cartilage at all on histology, which also is very distinctive and helps you figure out where you are in your respiratory tract. Another important thing you can look out for is smooth muscle layers. They're more prominent here because they come up on histology as this sort of pinkish color. Let's move on to the respiratory bronchioles. So the respiratory bronchioles, before that, we can have a look at the terminal bronchioles. And they're called terminal bronchioles because they form the very end of the conducting system, the conducting zone. So they're the final branches of our conducting bronchioles. And again, they've still got the cuboidal cell type, so low cuboidal. So again, if you remember, we started off pseudostratified all the way in the trachea. And then as we moved down, we went to cuboidal. And now notice, as we come to the respiratory bronchiole, which is the actual respiratory zone responsible for gas transfer, we've now got squamous cell types, and that's going to be responsible for conducting gases across, exchanging gases across it, such as oxygen, carbon dioxide, and other gases, which are going to play an important role in perfusion. All right, let's move past this and take a look at our pneumocytes. So if we go back here, the pneumocytes are the actual surface epithelial cells within the alveoli. So they form the cells that line the surfaces of the air sacs, the alveoli. And they come in two varieties, and they're very thankfully named, very simply named, type 1 and type 2 pneumocytes. Type 1 are these thin, simple squamous cell types with very evident nuclei, and they are going to form the barrier or one of the layers for gas exchange, whereas type 2 cells or type 2 pneumocytes are more larger cuboidal cells, and we're going to see this and appreciate this better on a diagram later on, which are larger, cuboidal, and they produce surfactant, which is their primary role. And we're going to see why surfactant is so important, both in this video, but also if you have a look at the breathing biomechanics video, we touch on very briefly how surfactant plays a role in lung compliance. All right, so now we come to the respiratory blood-gas barrier. So this refers to how gases are actually exchanged across the alveolar membrane. Now, if you look at oxygen, for example, which is absorbed into the bloodstream, you can notice that it has to pass through quite a few layers before it can actually reach our red blood cells and oxygenate them. So it has to, first of all, travel across a layer of surfactant. And if you remember, the surfactant was secreted by the type 2 pneumocytes. So that's your first layer, surfactant. And then it's going to meet our type 1 pneumocytes. So that's your simple squamous cells. And then once it passes through that, it's going to reach a basal lamina, because if you notice, as you know, epithelium has a basal lamina, so it's going to have to pass through that. And then it'll reach the endothelium, so the epithelial cells of the actual blood vessels, the capillaries. And then it'll enter the blood plasma, right? The fluid component of the blood, basically just the bloodstream, essentially. And then from there, it can get absorbed into the red blood cell. Technically, you can break that up into the red blood cell membrane and then crossing that into the red blood cell. But either way, it has to pass through all these different layers before it can finally reach your red blood cell and oxygenate it. So it's important to understand these different layers and where the cells sit in these layers. Note that type 2 pneumocytes, they exist in the respiratory epithelium, but oxygen doesn't cross them. Instead, type 2 pneumocytes secrete a chemical called surfactant, which the oxygen is going to be crossing. And you can imagine carbon dioxide does the exact same thing, but in reverse. So it passes through out of your red blood cells into the blood plasma, across the capillary epithelium or endothelium, across the basal membrane, and across the type 1 pneumocytes, through the surfactant, and then finally into your alveolar space. So with all that being said, we're going to briefly touch on a couple of small nitty-gritty points. First of all, surfactant, we already talked about how it's produced by your type 2 alveolar cells, which were those simple cuboidal cell types. And in terms of what it's composed of, it's about 90% lipid and 10% protein. Its primary function, as we talked about earlier, is to actually lower surface tension at the air-liquid interface, therefore making the lungs more compliant and easier to expand. And without it, we would actually have alveolar collapse. So it's really essential that we have surfactant. It also has a smaller role of helping maintain sterility and promoting immune function as well. And here is a useful summary of everything we've covered. Now, I know it can be a little bit daunting looking at histology and having to memorize all these different facts about it. But I think if you break it up into the general patterns that you observe, so for example, we would have already noticed that when it came to the conducting zone higher up, there was mainly ciliated pseudostratified columnar epithelium in the trachea and the bronchi. Then it transitioned into cuboidal in the bronchioles. And as we made our way to the alveoli, it became more simple squamous with the type 1 pneumocytes. So you can sort of look at those patterns and try and use those to help you remember how things transition. And then also looking at things like the cartilage and how it goes from being present to patchy and then absent. And then smooth muscle as well starts becoming more prominent and then disappears in the sort of respiratory zone. All right, so it's also important to keep in mind a couple of numbers. The only reason I include this is not because I think they're particularly useful, but because I do recall in my notes writing down that these numbers were something recommended by faculty to memorize for the purposes of your classes and even the exam. So just have a look at these numbers, see if you can memorize them. They're not too difficult to remember, but just keep them in mind. So the first one is that number of alveoli in each lung is about 150 million. So that's in each lung. And that's on average. And the surface area of the alveolar membrane is about 70 meters squared, which they like to include the fact is that it's roughly equivalent to the badminton court surface area. And the thickness of the alveolar membrane, so that membrane that we had a look at earlier, if we have a look on the previous slide, that membrane is about 0.2 micrometers. So it's extremely small, but it does have a thickness of 0.2 micrometers and plays an incredibly important functional role. All right, and that is it for respiratory histology. Again, if you have any questions, concerns, feedback at all, let us know in the comments. And we'll see you in the next one.